Good morning, everybody. Welcome to another edition of Lab 207 webcast. My name is Mr. Kite, and I'll be hanging out with you today as we are nearing the end of our series on populations. Topic for the day is going to be toxicology, so like always, let me get you some objectives and we'll get going. By the end of this video, 4 things I need you to be able to do. First one, describe each of the major categories of chemicals. Second, understand the purpose of an LD50 study. Third, explain the factors that determine the concentration of chemicals in organisms. Guess there's 3 things I need you to do today. So, could be a longish video. Let me jump in and start talking. In our previous video, we talked about things that can affect the lifespan of a human. Mostly we talked about diseases. Today, we're going to talk about chemicals because chemicals can be just as, if not more, harmful to the human body and the human lifespan as a pathogen. So first thing I'm going to do is I'm going to go through some common categories of chemicals, and we're going to talk about the effects of them and where they come from. So for each of these categories of chemicals, you need to know an example, what they do, where they come from. First category that you need to know is going to be a neurotoxin. Now, name pretty much gives this one away. Neurotoxins affect the nervous system. Also, they generally impact the kidneys. For the most part, these are going to impair nervous function. They might kill off neurons, some sort of damage to the nervous system. Some examples of these, the ones that you would definitely need to know, would be insecticides, lead, and mercury. So those would be your examples. Also know that a lot of snake poisons are neurotoxins. So that would be your first category of chemical. Second category of chemical is going to be a carcinogen. Over there on the right, you see a micrograph of a chemical called asbestos. It is an example of a carcinogen that is inhaled. Carcinogens as a group cause cancer. There's a type of carcinogen known as a mutagen. Mutagens damage DNA. So not all carcinogens are mutagens. All carcinogens cause cancer, though. Examples that you would need to know would be asbestos, which I just mentioned, used to be in insulation and things like that, and it'd be breathed into the lungs. There's radon gas that comes from the decay of uranium in bedrock. There's formaldehyde, which is a preservative. Those would be the major carcinogens you need to know. Next up would be a teratogen. Teratogens are chemicals that impact the development of embryo and fetuses when they're within the womb of a mother. Teratogens lead to birth defects, and the 2 common ones that you need to know is thalidomide, which was a drug that was given to pregnant women to reduce the impacts of morning sickness. This was in the late 50s, early 60s, and it led to really significant birth defects. And then there's alcohol, which you've heard about leads to fetal alcohol syndrome. Next up on the hit parade is an allergen. This is probably the most common chemical that damages human health. This is going to be any chemical that causes some sort of allergic response, usually associated with breathing difficulties, rashes, itching, watery eyes, things like that. Allergens are going to be unique to a person. So I, for example, am allergic to some pollens, which you see there on the right. My wife is not. Some people are allergic to cats, dogs, peanuts. You name it, there is probably a person who is allergic to it. One of the scariest ones I guess you need to know about is an endocrine disruptor. And so just a little bit of background. Hormones are responsible for signaling within our body. They're released by the brain and a couple other organs. They travel throughout the bloodstream. They attach to receptors on specific cells, and they cause some sort of action in that cell. Endocrine disruptors disrupt that signaling pathway. And the big thing that I need you to know about endocrine disruptors is that they generally are released into the water supply. 2 biggest sources would be animal rearing facilities, so places where they raise a lot of cattle or chickens or things like that, because the antibiotics and the hormones that they give those animals make it into the water supply. Also, human birth control pills contribute to a large amount of endocrine disruptor in the water supply, because as women take birth control, it's metabolized, and then as they urinate, it goes into the toilet and then down into the sewer. Endocrine disruptors have been strongly associated with feminization of aquatic animals, particularly fish, reptiles, and amphibians. Animals that have been exposed to endocrine disruptors may show, like males exhibiting female characteristics such as testes that produce eggs, converting from male to female, producing sperm that are incapable of swimming. They may be sterilized, so these can mess with the reproductive system of reptiles, fish, and amphibians. It's also been shown that there may be similar impact on humans. All right, next thing I need you guys to know about is going to be a dose response study. And basically, I just went through all these categories of chemicals that can potentially harm the health of humans and other animals. But scientists, obviously, as they're making chemicals in the lab, need to know, all right, how much of this chemical is actually dangerous? What is the dosage that an animal can be exposed to before it becomes a problem? So what they do is dose response studies, and they'll choose like a representative animal for each like category. So there might be like a water flea for insects and a trout for fish and maybe a pigeon for birds and maybe a rat for mammals. Obviously we can't do these kind of studies on humans, so the results from those other organisms are extrapolated out. But basically what they do is they expose animals to increasing concentrations of whatever the substance is and record what percentage of the organisms tested die off. So down here you can see when doses of the chemical are low, not many organisms die off. As that dose reaches a critical point, a bunch of your organisms are going to start dying off very quickly. When you get right here, this is called the LD50. This is the point at which 50% of the organisms die. Lethal dose 50, meaning 50%. So a lot of chemicals will be tested for their LD50. What is the dose of that chemical that kills off 50% of the organisms? And then once you pass the LD50, organisms die off very rapidly until you get to 100% mortality. So this is the basic method that scientists use to determine the toxicity of a chemical, and then they can, once they've figured out the LD50, do comparisons based on known chemicals to say, all right, is this new chemical more or less toxic than chemicals that we already know about? As far as regulation goes, throughout this class we'll talk about regulation here and there. When we're talking about chemicals, two major pieces of legislation you need to know. There is the Toxic Substances Control Act, which controls most industrial chemicals, and then there's the for pesticides there's the Insecticide, Fungicide, and Rodenticide Act. Both of those regulate the use of chemicals, and they determine safe levels of chemical that can be released into the environment or detected in the environment. So know those two sets of regulation. And we're going to move forward into humans. How do we determine what's okay for a human? So obviously we can't do an LD50 study on humans. So generally what they'll do for humans is they will take the concentration that is safe for a rodent, like a rat or mouse, and then divide that concentration by 1,000, because they figure rodents and mice may be more resistant to chemicals than humans, and also just to be safe they throw another division in there. So safety for humans is usually whatever the LD50 is for a mouse or a rat, and then divided by 1,000. Let's talk about studies as we kind of move on towards the end of our talk today. So there are two major types of studies that scientists carry out with regard to chemicals that you need to know about. The first one is a retrospective study, and a retrospective study basically takes a group of people that we know have been exposed to a chemical, and you look at what the chemical did to those people, and then you track the people going forward that survived to see what happens to them. An example of this is in 1984, there was a chemical accident in Bhopal, India, where a toxic gas was released from a factory into the city. It killed 2,000 people immediately, 15,000 over the coming year, and then over 100,000 people were sickened going forward. So that would be an example of a retrospective study. They saw something happen, they looked at the results, and then they tracked the people going forward. You can contrast that with a prospective study, which is basically looking at a group of individuals that you know might be exposed to a chemical and comparing them with a group of people that you know probably won't be exposed to a chemical. So an example of tracking people going forward would be studying lead. Scientists know certain populations of people might be exposed to lead. So over the course of those people's lives, they will track their exposure to the foods, the places they live, the chemicals they interact with, and see what happens to those people over time, trying to determine the impacts of a particular chemical. Now, prospective studies are difficult because they can include synergistic interactions, which is essentially where multiple factors interact to cause a result within the body, and that obviously makes it hard for a researcher to say, this is the thing that is causing the problem. It would be hard to say lead is causing this problem because there might be other chemicals interacting, causing the same results. So retrospective studies versus prospective studies. And what we're going to finish up with today is going to be determining the concentration of a chemical that a person might be exposed to and how much can be, kind of concentrated in their body. And there's four things that we look at when we're talking about concentrations of chemicals in a person. So the first one is the route of exposure, and depending on the chemical, there are a bunch of ways that a person could be exposed. Some examples or possibilities is going to be soil. So that could be from the vegetables that are grown in the soil or just soil contamination and things that you eat. Could be in the food that you eat, could be in the air you breathe, or could be in the water you drink or bathe or swim in. So depending on the route of exposure, the concentration could be higher or lower. The next important thing that we look at is the solubility. Different chemicals dissolve in different substances at different rates. So some chemicals are highly water soluble, which means that if they are released into a water supply, they will dissolve into that water supply very easily, and then they will be carried along with that water. Others aren't so soluble in water, but they might be soluble in fats or oils. And chemicals that are really soluble in fats and oils might hang out in soil really well. Also, chemicals that are soluble in fats and oils accumulate in the body very well because a lot of these cell membranes in our bodies are made out of fats and oils. So chemicals that dissolve in fats and oils can hang out in our bodies for quite a while. Two things left. Biomagnification is something that I am betting the AP is going to ask you about. It also talks about concentration. This is basically the idea that the concentration of a chemical within the body of an organism increases as you go up a food chain. And there's a really easy way to illustrate this. So there's a pesticide called DDT. Really effective pesticide, but it has some pretty bad effects on birds of prey. And here's essentially how it works. If DDT gets into water, you get these little plankton, and the plankton get the DDT into their little bodies, which for them is not a big deal because they've just absorbed a little bit of DDT, probably not enough to cause them a problem. Let's say each one of them absorbs one particle of DDT. They're, probably doing okay. Going up a level on the food chain, you have got a stickleback. Now let's say that this stickleback needs to eat 10 plankton in order to survive. So each of these plankton just got one particle of DDT. If he eats 10 of those plankton, then he gets 10 particles of DDT. Now we're going to go up here to our flounder. Let's say that our flounder needs to eat 10 stickleback in order to survive. So every one of those stickleback he eats, he gets 10 particles of DDT. So this means that in our flounder, we are now up to 100 particles of DDT. And then our osprey up here, our bird of prey, let's say that he needs to eat 10 flounder. So when our osprey eats our 10 flounder, each time he eats a flounder, he gets 100 particles of DDT, which means that our osprey gets 1,000 particles of DDT. And DDT has been found to weaken the eggshells of birds of prey, which means that when they sit on them to incubate them, they crush the egg and kill the baby. This is one of the reasons that DDT was it was outlawed in America because of the effects on birds. Last slide for the day is going to be persistence. Persistence essentially looks at how long does a chemical hang out in the environment and remain active. Some chemicals are highly persistent, others aren't. There are some pesticides that break up as soon as the sun comes out. Others, like DDT, highly persistent. It can hang out in the environment for 30 years. So all of those things that I just went through, the route of exposure, the solubility, biomagnification, and persistence, will help to determine how much of a chemical a person is exposed to and how long it hangs out in their bodies. And that's it. Thanks for joining us on the Lab 207 webcast. My name is Mr. Kite, and hopefully we'll see you again.